In part two of lab two, we'll consider the membranous organelles. If you recall from part one, we looked at non-membranous organelles. So the difference here is membranous organelles, each of the organelles has its own membrane, right, that separates the organelle from the cytosol. Let's take a look at the nucleus first. So the nucleus is usually located somewhere in the middle of the cell. We will do some exceptions such as adipose cells, muscle cells, skeletal muscle cells, where the nucleus is not in the middle and it's going to be pushed off to the side. But for the most part, think of the nucleus as being in the center. So, same as our brain is the control center of our body. The nucleus is similar to the brain. Just kind of think of it that way. It kind of controls everything. It's responsible for protein synthesis. This is where our genes are. It's responsible for cell division and even controlling metabolism, the metabolic rate. So it's really controlling everything. Here's some of the parts that you need to know. So this white membrane right out over here, this is the cell membrane going around, or the membrane going around the nucleus. This is known as the nuclear envelope. It's a double membrane, and you can see here it's got some large holes inside of it. All of these holes are known as the nuclear pores. So as materials are made inside the nucleus, right, they actually have to go through the pore and then come out over here. An example would be the ribosomes. In part one, we looked at the ribosomes. They're made here in the nucleolus. The ribosomes then have to get out. They go through the nuclear pore and they come out this way. Some other things would be like messenger RNA, also created inside here, inside the nucleus, and then it goes through the nuclear pore and then out to the ribosomes. So nuclear envelope is the membrane that encloses the nucleus. The nuclear pores are the large holes. Now, the cytoplasm within the nucleus has a specific name. It's called the nucleoplasm. Nucleoplasm. So this has all different enzymes and materials that the nucleus needs to function. We even see inside here these little blue coils are representing the DNA. Now, this structure right here, the nucleolus, is kind of like a pit. It's right in the middle, and for the most part, anything that's involved with protein production is going to be produced here in the nucleolus. Okay, one other thing I just noticed I have here in the diagram for you, we call the DNA chromatin. In a non-dividing cell, or a cell that's not going through cellular division, the DNA doesn't show up under the microscope. We can't see it. It's referred to as chromatin. Once we look at cell division and the cell is actively dividing, the same DNA looks like chromosomes. So in a dividing cell, the DNA is called chromosomes. That's just a new term that we haven't used yet. So again, nuclear envelope, nuclear pore, nucleoplasm, nucleolus, DNA, or chromatin. I may have put in your notes nucleoli. That would just be plural. Sometimes it could be two nucleoli. Nucleolus would be singular. So here's again the picture from our book. You can see that the nucleus is pretty much in the center over here, and this would be a blow up of it. Same structures, right? Nuclear envelope, nuclear pores, but you can see that right there. Here's the nucleoplasm, nucleolus. Our second membranous organelle is going to be the endoplasmic reticulum. So we can divide the endoplasmic reticulum into two types. If you recall from part one, sometimes ribosomes are attached to the endoplasmic reticulum. So these white dots right here, just as a review, are the ribosomes. When they're attached to the endoplasmic reticulum, we call that the rough endoplasmic reticulum. Right? It kind of looks like sandpaper, so it looks rough. Just think about this. If you remember from part one, the function of the ribosomes is to synthesize proteins. So that kind of gives you a hint here as to the function of the rough endoplasmic reticulum. Since it has ribosomes on it, right, this, you can actually think of this as a protein factory. Down over here, notice this endoplasmic reticulum does not have ribosomes on it. This is known as smooth ER, right? It's because it has more of a smooth appearance. Since it doesn't have ribosomes, the smooth ER is not going to be able to make proteins. Okay, so rough ER is going to have ribosomes. Now, don't let this confuse you. What they did here in the model company, they cut off some of the membrane so that we're looking into the rough ER. Right, so this is just like the insides of the rough ER. Same thing over here. And then here too, they cut off the membrane so it looks like this is smooth, but it's not. They just cut off the membrane with the ribosomes on it. We're actually seeing the channels within the rough ER. So don't let that throw you off. Let's take a look at what are some of the functions of the rough endoplasmic reticulum. So again, just the fact that it has ribosomes, it's going to be active in protein and even glycoprotein synthesis. A glycoprotein is a protein that has a carbohydrate. So that's going to be its major function. The smooth ER, since it doesn't have ribosomes, as I mentioned before, will not make proteins. Instead, it's going to do things like make the components of the cell membrane. Let's just say something needs to be repaired here in the cell membrane. The smooth ER can make phospholipids, it can make cholesterol. In certain cells, such as the testes or the ovaries, it makes steroid hormones. You may have heard of testosterone or estrogen. In some of the other cells, it'll make fats. Even in the liver has a lot of smooth ER. It's involved with detoxification, so it takes out any chemicals that may be damaging to us. Okay, so I always like to think of as the endoplasmic reticulum as a factory, right? They're factories that are producing different things. Again, if ribosomes are present, you know that they're producing proteins. If the factory here does not have ribosomes, as I mentioned earlier, it's going to produce the other things. So here's a picture from our book. If you notice, the ER is usually right next to the nucleus. Right? So here's the ER, here's the nucleus. Right? You can see they're usually in close proximity to one another. Here they're actually showing us the rough endoplasmic reticulum with the ribosomes, and then over here they're showing you smooth ER without the ribosomes. And a blow up of the same picture. Now, the next structure is known as the Golgi apparatus. I like to think of this as a warehouse. I like to think of the endoplasmic reticulum as a factory. So now the factory is going to send its materials up to the warehouse. The warehouse we're going to call the Golgi apparatus, and it's also just stacks of membranes that are folded on one another. They're going to be up over here. So all of this is the Golgi apparatus. Here's some of the things that the Golgi apparatus does. It modifies and packages different things that are made in the endoplasmic reticulum, and then it sends them out of the cell. We're going to talk about a process called exocytosis. This can also help to renew parts of the cell membrane that are damaged. Let's just say over here, if the cell membrane were damaged, the Golgi apparatus will send like one of these bubbles that are just a membrane that are empty, will send that bubble over to the cell membrane and repair this damaged cell membrane. Because again, this is just a bubble made out of cell membrane. And we'll talk about that in a little bit. And then the third function, the Golgi apparatus will produce these yellow things over here called lysosomes. We'll talk about them in a minute. These contain powerful digestive enzymes. All right, so the three functions, just in a general way, the three functions are going to be the Golgi apparatus will export materials via a process known as exocytosis. It can help to repair the cell membrane if it's damaged, and it's going to produce lysosomes. So I'm going to take you to a little bit of an advanced diagram just to kind of put this together for you. Over here, this is the nucleus. Right? The nucleus, we said earlier, kind of regulates protein synthesis. So once the nucleus decides we need to make a protein, it's going to it's going to send a message to the endoplasmic reticulum to start making a protein. Right? It does that by, and this is a little bit advanced, by a piece of messenger RNA. Right? So the nucleus is kind of notifying the endoplasmic reticulum with a messenger RNA. If you look at this endoplasmic reticulum, you're going to notice these blue dots are the ribosomes. Okay, so right off the bat, we know it's rough ER, and rough ER is always going to produce proteins. So what's happening, if you look here, inside the endoplasmic reticulum, all of these little balls right here are amino acids. Once we line up amino acids, that produces a protein. And now we have this protein. So here's the protein inside the ER. Right? So this is the factory. It just made a protein. You'll notice the protein works its way through the endoplasmic reticulum and eventually is going to end up in this little area here. It's almost like a water balloon, but instead of having water in it, it's going to have proteins in it. Eventually, this pinches off, and now we have this little balloon with protein inside of it. And if you notice here, it's called a transport vesicle. For lab, you don't have to know that. It's not on our model, but I'm just going to explain it since it's here. The transport vesicle is going to drive the protein up to the warehouse. All right, so again, this was the factory. The transport vesicle drives the protein to the warehouse. And notice the transport vesicle then blends with the Golgi apparatus. This area here where the protein enters is known as the forming phase. Again, you don't have to know that for lab. This is more of a lecture material. I just want to throw it out there since it's on here. And then the protein enters the Golgi apparatus. Right? It goes through the Golgi apparatus. The Golgi apparatus may modify and reorganize the protein a little bit. And if you look over here, now we have proteins on the other side of the Golgi apparatus. This is called the maturing phase. But take a look at the protein. See how it looks different here than when it came in, right? They look different. That's because the Golgi apparatus modified the protein to make it a functional protein. So let's look at the three functions of the Golgi apparatus. This picture does a good job with that. Let's take the exocytosis first. So now our protein is in this little balloon right here. Again, think of it as a water balloon, except it doesn't have water in it. It has a protein. This will pinch off. Then the vesicle, now called the secretory vesicle, will migrate like this to the cell membrane and blend with the cell membrane over here and open up, and it will actually release the protein from the secretory vesicle out into the extracellular fluid. So let's just say this was a hormone like insulin. This would then go into the extracellular fluid and then go into the bloodstream and do the function of insulin. So this process is called exocytosis. We could actually see here's a secretory vesicle kind of spitting out the protein via exocytosis. All right, so that's the first function of the Golgi apparatus. It's going to release proteins via these secretory vesicles. And this one I do want you to know, this is on our model by the process known as exocytosis. Over here we see another protein was made. This is a different protein. When this pinches off, this is not going to migrate to the cell membrane. This is actually going to stay in the cell, and this is going to be our lysosome, which we're going to talk about in a little bit. The third function here, notice this little bubble right here has nothing in it. It's just the membrane. Okay, and this is equivalent to the cell membrane. Let's just say a piece of cell membrane over here was damaged. Right? This will now migrate over to the cell membrane over here. You can see they call it a renewal vesicle, and it will replace what was damaged. It's almost like fixing a hole in your wall. If you had a hole in the wall, You could just put a new piece of sheetrock there. You could spackle it, you sand it and you paint it, and you never know there was a hole there. Similar type of thing. Okay, so again, the three functions of the Golgi apparatus is going to be exocytosis, right? Release materials from the cell via exocytosis, do cellular repair, cellular membrane repair, I should say, and then also form lysosomes. So let's go back to our model. So over here, let's say this would be the endoplasmic reticulum. It's a rough ER, so it's producing proteins. We wouldn't see it here, but the transport vesicles will drive the proteins to the forming face over here of the Golgi apparatus. So this is where the protein enters the Golgi apparatus, right? It goes through the Golgi apparatus, it gets modified. Now, these bubbles right over here, guys, all of these and these are the secretory vesicles. So let's just say this secretory vesicle here has a protein in it. It's going to kind of drive over here. Now we see it again. It's going to release the protein right here via that process known as exocytosis. So I labeled this here as an exocytotic vesicle, right? The secretory vesicle. Once it comes over here and it's doing exocytosis, we could call it an exocytotic vesicle. Right? Golgi apparatus, secretory vesicles, and then they form the exocytotic vesicle. And here's a picture from your book. You can see just here's the ER, here's the Golgi apparatus. This would be the forming face, and here's all the folds right here. This would be the maturing face, and then these would be the secretory vesicles right here. And you can see here's a secretory vesicle actually blending with the cell membrane, and this is where exocytosis would take place. Again, just a blow up. Okay, so the next organelle, most people know this one. They kind of remember this one from like a high school biology course, is the mitochondria. When we think of the mitochondria, a lot of times people say it's the powerhouse of the cell. And that is absolutely true because this is where we produce most of our ATP. I would say like 95% of our ATP is produced in the mitochondria. So on this model, all of these orange structures, here's a mitochondria. Now this one is cut, so we're looking into it. Let's say this one here, this one is not cut. We're seeing the whole mitochondria. So all of these orange structures would be the mitochondria. Again, if you notice, it has a cell membrane. But then there's also cell membranes in the middle. These folds right here are also cell membranes. These we call cristae. This is where the chemical reactions take place to produce the ATP. If you've ever heard of the Krebs cycle, also known as the citric acid cycle, oxidative phosphorylation. This is where we actually generate the ATP. Okay, so those are the mitochondria. Again, here's a picture from our book. You can see again these mitochondria, excuse me, these mitochondria are cut, so we can see the inside of it. Here's one that's partially cut, partially intact. Again, here's a blow up. Again, here's the outer membrane. Here are those inner folds, right? These are called the cristae. The fluids that would be inside here, they refer to as the matrix. But this is right here, the matrix and the cristae is where all the chemical reactions are taking place to form the ATP. Here's an actual mitochondria. Look at this. They had to magnify this 46,000 times to see the mitochondria. In our lab, the best that our microscopes do is 1,000 times. So we would never be able to see the mitochondria just looking at a cell in the lab. Our microscopes aren't that strong. And this is just an overview slide for lab. You don't have to worry about this too much. We're going to get this into lecture. But here you can see glucose being converted into pyruvic acid. There's a 10 steps of a chemical process known as glycolysis. But this pyruvate or pyruvic acid then enters the mitochondria. Right? It goes through a series of cycles here known as the citric acid cycle, and then it goes into something known as oxidative phosphorylation, and the end result is we produce ATP. And that's the main thing I want you to know for lab. If I were to ask you what's the function of the mitochondria, you can say powerhouse, You can say ATP production, produces ATP, however you term it, as long as you get that idea. The last two organelles I kind of think of as part of the digestive system of the cell. This is going to be the lysosomes and the peroxisomes. We also had considered, just to remind you, in part one, the proteosomes, which I kind of call the digestive system too, but that's a non-membranous organelle. That's why I had done it previously. And if you remember, the proteosomes broke down and disassembled abnormal proteins. So it's a way of getting rid of abnormal proteins. The lysosomes are in this lab, part two, because they have a membrane. So I labeled them here for you. All of these yellow structures here, these yellow membranes, are going to be the lysosomes. Here's a lysosome that's cut, so we're looking into it. That's why the color looks a little different on the inside. Here's the lysosome intact. So one of the main things that the lysosomes do is that they can get rid of worn out organelles. We actually call that autophagy. I didn't put that in your notes. I just kind of throw it out there if you ever hear it. Let's say, hypothetically, this mitochondria here stops functioning. It's done its time, right? It's kind of worn out. So the lysosome can come over and attach to the mitochondria and release its enzymes into the mitochondria, destroying the mitochondria. Good news is we can make new mitochondria. They actually can replicate, so we can replace any worn out mitochondria or other organelles. So that's one thing. The other thing is sometimes we have extra cells. We make extra cells, and if we don't need them, the cell can be destroyed. So the lysosomes will release their enzymes in the cell and kind of, it's almost like committing suicide, kill the cell. This process is known as autolysis. Autolysis. Okay, even white blood cells, let's say they have lysosomes. Let's say the white blood cell ingests a bacteria. The bacteria will be broken down by the lysosome. The last membranous organelle is the peroxisome. So on our model, if you look on the outside here, it's kind of grayish in color. Here's one that's cut, but if you look at the outside, it's gray. I mean, here's another one over here. It's gray. But then when we cut it, it looks a little bit kind of purplish or violet. These are also contain digestive enzymes. However, they break down organic compounds. They're not going to break down organelles or destroy the cell. They're going to break down organic molecules such as like fatty acids. They can also be involved with detoxifying some harmful substances. Okay, so those are the peroxisomes. All right, so those are all of our membranous organelles.